Today we're talking about how things move in and out of a cell membrane. Sometimes you need to send materials against a concentration gradient. So if you look at these purple squares, there's a low concentration of them here. And here you have a bunch of them. That's a high concentration. And so occasionally, or maybe even often, you need to move materials against a concentration gradient. You would use active transport. Usually you require ATP for that. You always require some energy source. It's usually ATP. And you call that a protein pump. So the protein pump refers to the fact that this molecule here is made out of a protein. It doesn't mean you're moving a protein. You might be moving a sodium ion or sugar or whatever, but it's a protein pump. So it moves materials from a low concentration to a high concentration, which means against a concentration gradient. So a gradient is when there's a difference. Concentration gradient is when there's a difference in concentration. If you're going against a concentration gradient, think of the normal flow as being from where there's a lot of them to where there's not so many of them. You're going backwards from that. So if there's a water going down a hill toward you and you're walking in this maybe shallow water, if you're going upstream, you're going against the concentration gradient. So you might also say up a concentration gradient. So this requires energy. Usually it's in the form of ATP. We'll do a unit on ATP later. There might be another energy source, so we'll talk about that at the end of this video. So here it is. Against or up a concentration gradient means the same thing, high to low. So that would be like opening up a perfume bottle. Let's say there's perfume in here. It's in liquid form, and some of it has already moved out. Active transport would be taking these and shoving them back in. So in the world that we're thinking about, it's not something you can do, at least not for this example, not very easily, but you do it in your body all the time. So whenever you contract a muscle, calcium's released. To put it all back into the endoplasmic reticulum, it requires energy. So you would use a protein pump for that. So I want to talk a little bit about kinetic versus potential energy. If you've taken physics, you've done this before. If you haven't, even in elementary school, I think you've talked about this. So kinetic energy is energy of motion, and potential energy is energy that is based on like where it is gravitationally or elastic energy, but it's some energy that could be used. And the thing I want you to know is that you can convert back and forth between these, right? So like if you're a kid swinging on a swing, you're maybe sitting on this swing, wee, and you keep going back and forth, right? So at the top, you've got a lot of gravitational potential energy. As you're moving and you're at the lowest point, you don't have as much gravitational potential energy, but you're moving, so that's kinetic energy. Then you go up to the top of the swing again at the higher point, and you've got more potential energy, but you stopped for just a fraction of a second, and so that would be no kinetic energy. So you can convert back and forth between potential and kinetic energy. In this example, the water up here has gravitational potential energy, even though it's not really doing anything. And then when it falls over the waterfall, it's losing gravitational potential energy, but gaining kinetic energy, energy of motion. You could put a water wheel here and it could spin, and you could use that energy to do a lot of different things. One of them would be to convert that energy into electricity. Here's another one. Here's something that's burning. So maybe you've got wood in here or something, or it's a fossil fuel. So maybe there's coal in here, gas, something like that. And there's a chemical reaction, fire. So you're converting the potential energy of the chemical bonds in the coal into thermal energy, right? That's the sort of energy conversion from potential to thermal energy. And actually, there's also light, right? So light energy. And then here's an example of electrical energy. So here's a battery, and well, sort of a battery, and here's a circuit. So there's a wire connecting a light bulb to the negative terminal, and then you have these chemical reactions in here and the positive terminal, and it's showing electrons that can go kind of moving this way. The circuit would actually be going the other way, but anyway, that's potential energy to light energy, right? So this is electrical potential energy that's converted to light energy. So the thing I want you to know is that energy can be converted. So let's we'll be talking about that when we talk about these protein pumps and how energy can be stored sometimes inside membranes. So just to sort of orient you as to where we are, we're talking about active transport. Previously, we've talked about diffusion. So passive transport is when you have no energy required and you're going from a high concentration to a low concentration. If you're going right through the lipid bilayer, we're going to call that simple diffusion. If you require a protein like here and here, that's facilitated diffusion. But if you're going from a high concentration to a low concentration, we're going to call that passive transport or a type of diffusion. If you're not, let's say you're taking these squares and you're shoving them to where there's a lot more squares. You're going against the concentration gradient. And if you're taking these circles and you're shoving them this way, you're going from sort of, do you see how it's backwards? The squares are going down and the circles are going up, or the squares are going into the cell and the circles are going out of the cell. But for both of them, they're going against their concentration gradient because the circles are going from where there's not many of them to where there's a lot of them, right? So for the circles, here's the high concentration. And for the squares, here's the high concentration. So to do that requires energy, and usually that's in the form of ATP. And so that's what we're talking about today. The materials that you could move this way are the same as the materials that would move through facilitated diffusion. So ions and any larger polar material. If you were shoving oxygen against its concentration gradient, it would be a massive waste of energy because it would just diffuse back through the lipid bilayer. But because you can control ions, sugars, amino acids, that kind of thing, you can put energy into shoving them in a certain direction and knowing that they'll stay there unless you allow them to go through facilitated diffusion through another protein. Okay, so ion pumps maintain membrane potential. So let's talk about this a little bit. So membrane potential means potential energy. And the way we define that is voltage. So voltage is a potential energy. It's a difference across anything. So it could be the difference across a circuit. So for example, here's a light bulb and the circuit goes from this source. So this could be a battery or in any case, it's a source of electricity. And the electrons are going through this whole circuit or through this other circuit. And you're converting electrical energy to light. And there might be some heat associated with this too, but you're doing an energy conversion. So that voltage is a potential energy difference. So it's a potential difference from here to here. That would be the voltage. And if you connected these electrodes from here to here, you would see a voltage drop as it goes over the light. But what you need to know for me isn't all this stuff. I want you to know that membrane potential is really a voltage across a membrane. So let's look at it in a cell. This is a neuron. And a neuron is found in your brain and spinal cord. It's also found going from your senses, like your skin, into your spinal cord or brain, or going out from your brain and spinal cord to your muscles. So the neurons in your body regulate at a very, very fast speed, regulate the body. So notice if you take this neuron and you're looking just at a little piece of the neuron, the axon is the part that takes the impulses toward the end. So if you look at that, that's right here. This is a cross section of the axon. And notice you've got these positive charges outside. Those are caused by sodium. So normally the cell has a whole bunch of sodium ions. I'm going to draw them in red. Whole gazillion sodiums out here. And then inside, you've got potassium ions, which are positive, which is kind of weird. I'm going to draw those potassiums inside. Kind of weird because I'm saying this is negative, but it really is for a couple of reasons. One is that there's also some other like proteins that are negative. So you've got other ions in here besides potassium, and they're negative. Remember, potassium is with a K, not a P. That would be phosphorus, and sodium is Na, not S, because that would be sulfur. So anyway, potassium, oops, potassium and sodium are two ions that I want you to know. Okay, so when an impulse goes along a neuron, it starts maybe here and it goes along this way. And when it gets right here, what happens is that the sodium ions diffuse in. They're going just with facilitated diffusion, high to low. They're not all going in, but some of them are going to go in. And the potassium I think I picked the wrong blue. The potassium, some of those are going to diffuse out. And so what I want you to realize is that these ions end up in the wrong place. And so what you need to do is use active transport to shove them both back out. So you're going to use active transport. Whoops. You're going to use active transport to shove your sodiums back out, and you're going to use active transport to shove all of your potassiums back in. And that's going to be a voltage difference of negative 70 millivolts. So milli is 10 to the negative sixth. This is a tiny, tiny little difference between outside the cell and inside the cell, but it's enough so that you're actually electrical. So your neurons all work like this and your muscle cells all work like this. And you know you're electrical because if you've ever watched TV and watched an EKG being performed, they'll do that on various TV shows, your heart rate will go, you'll see the heart trace, right? Beep, beep. I'm doing kind of a bad job drawing it, but this is your sodium and potassium moving in the top of your heart, the ventricles, I mean the atria, and then the bottom of your heart, the ventricles. So you know you're electrical, right? And if you ever have seen an EEG, that's of the brain, and you'll see a bunch of electrical activity in there. And that's all of the sodiums and potassiums moving in all the neurons in your brain and in that area. All right, so vocab word for you to memorize, electrochemical gradient. So a gradient is a difference between one place and another. So notice here the extracellular fluid that's outside the cell, right? And the cytoplasm right here that's inside the cell. Notice that there's a lot more hydrogen ions right here. So we should talk about hydrogen a little bit. Where's my pen? Right here. Okay, so hydrogen. If you remember the periodic chart, hydrogen has one proton and one electron. So sometimes it will lose that electron. Whoops. And when it loses, oh no, what did I do? Okay, let's do hydrogen. So hydrogen has a proton and an electron. And sometimes, I think I have to move myself out of the way. Sometimes that electron will be lost. And when it is, what you have now isn't a hydrogen ion anymore. Isn't a hydrogen atom. Now it's a hydrogen ion. But notice, since it's hydrogen with only the one proton, you can also just call it a proton. So a proton is actually the same thing as a hydrogen ion. So this protein right here that moves stuff, this protein pump, it's a protein pump because it's made out of protein. It's a proton pump because it's moving protons, or you could call it a hydrogen ion pump. It's shoving hydrogen from a low concentration, there's only one of them here, to look at the bunch of hydrogens, you've got a high concentration over here. So you're shoving the hydrogens against their concentration gradient, and it takes energy to do that. So an electrogenic pump is a transport protein that generates voltage across the membrane. So voltage is a difference electrically between the inside and the outside. And so here's something kind of cool. In animals, it's a sodium-potassium pump. So our nerve cells and our muscle cells both work the way I showed you. Oh, I skipped a page. The sodium-potassium pump is going to be a proton. It's going to be a protein that's right here that shoves sodiums back out. They might have diffused in, so it'll shove the sodiums back out, all of them, and it'll shove the potassiums back in. And so both of them will be going against their concentration gradient, but in different directions. So that's going to be a sodium potassium pump. And that sets up this voltage. It's a difference between the inside and the outside. And because you have some negatives, some negative ions inside, and because the sodium tends to diffuse a little bit at a little bit different rate than the potassium, you end up having the inside mainly negative, and the outside generally positive. And so that gives you this voltage difference, this difference in electricity. So electrochemical gradient, that's when you have a chemical difference, so a chemical force, meaning this is the hydrogen atom that's lost an electron, right? So this is a different atom, really, than you have a different number of hydrogen ions here than you do here. That's a chemical difference. And then there's also an electrical difference because it's charged. So out here you tend to be positive, and in here you tend to be less positive, right? So an electrochemical gradient is a difference between the inside and the outside, both chemically because it's a different atom, and electrically because it's charged, it's an ion. So, so here's the thing that's kind of cool. This proton pump exists in bacteria and fungi and plants, and ready for this? Also in mitochondria. Cool, right? Oops. Mitochondria and chloroplasts. More evidence, right, that maybe they evolved from bacteria. And in animal cells like muscles and nerves, sodium-potassium pump. All right, so these store energy that can be used later for work. So this ends up being kind of like a battery. You know how with a battery, you've got a negative side and a positive side, and if you hook this up to a light bulb, and you connect it there, and let's finish drawing. Whoops, the light bulb should look kind of like this. Anyway, here's this battery, which is really potential energy, right? Chemical, electrical potential energy. And then you hook it up to a circuit with a wire and a light bulb, and you can convert that electrical energy into light. Same thing with this. Having all of these, a high concentration of hydrogen ions in one spot, or of anything in one spot, they're naturally going to end up going the other way by entropy, right? Just by things spreading out. And so these pumps set up a gradient, and that's potential energy. All these hydrogen ions over here is really both electrical and chemical potential energy, because it's a gradient. It's a difference. Just like there's a gradient between this side of the battery and that side of the battery, and that's what makes the light go. Just like there's a gradient between water that's at the top of a hill and here's the water flowing down a water flow. So there's this gradient between like a gravitational difference, right, between the top and the bottom there. All right, so last thing I want to talk about here is CoTransport. So usually active transport requires ATP. We will do a unit on ATP later, but generally the energy source that cells use is ATP. Here's a case where it's not. There's ATP at first, but then there's something else that's used. So let's say, so co, first of all, would mean there's two things going on. So like a co-teacher or co-captains, there's two of them involved, right? So CoTransport involves two different molecules or two different materials. So what's happening here is that you've got ATP, so energy in the form of ATP, and that's used to shove this hydrogen ion against its concentration gradient. Notice there's a low concentration of hydrogens here, but a high concentration of hydrogens here. So you're shoving the hydrogens this way, which is setting up a gradient, an electrochemical gradient. So the ATP is used to set up this gradient, and now you have a difference, right? Just like when you have a battery with a positive side and a negative side, that's a gradient. That's a difference between one side and the other side in terms of electricity. And that's really what you've set up here. This side is mainly positive, and this side is more negative. So that's just like a tiny little battery. So now you have this high concentration of hydrogen ions. They're going to go through this way by facilitated diffusion. Can you see that? I think I'll change color. So the proton pump, this is active transport, right? Because you're shoving stuff against its concentration gradient. When the hydrogen ions go out with their concentration gradient, that's just facilitated diffusion. It's going from high to low. That's the normal way. That's really potential energy over here, right? That's being released as kinetic energy. And when that energy is released, it's used to take sucrose, sugar, and shove it against its concentration gradient. So this part of the transport would actually be active transport. So against concentration gradient. And so this part, what's happening here, is active transport. And so that's this part of the transporter is really a protein pump. So it's physically made out of a protein, and it's pumping sugar across the across the membrane. So CoTransport occurs when active transport of a solute indirectly drives transport of other solutes. So you're actively transporting hydrogen this way, and then as it comes back, you're actively transporting sucrose that way. So it's really an indirect thing because here's one protein pump that's transporting protons, right? And then that's used as it comes back this way to shove the sucrose the other way. And that, I think, is my story.